Good evening, everyone. Welcome back to Living Systems, and we are in week 9 of semester, week 9 of 12. So we're really at the business end. I can see all those assessments start to stream in for various subjects. So that's really great to see. So tonight we're really continuing on from our lecture on cells, on functioning cells, but we're going to take a closer look tonight at membranes, biological membranes, and how substances move across membranes. So let's make a start, and we're going to really focus on plasma membranes. So remember, the plasma membrane is that lipid bilayer that surrounds every cell, regardless of whether we're talking about plant cells, animal cells, bacterial cells, and so on. And of course, you know that plasma membranes control the passage of substances into and out of a cell. And they do this to maintain stable conditions inside the cell, which is termed homeostasis. And we can see a great example here, which provides some evidence of how selective plasma membranes are. So this is a typical animal cell, a blood cell, and we can see the concentration of certain ions within the cell. So potassium, 140 inside, 140 millimolar inside, sodium ions 6, chloride ions 8 millimolar inside the cell. But outside the cell, in the blood plasma, the concentrations are very different. And you can see they're sitting here at 10, 135 and 110 millimolar. So this is already evidence that the cells can control and do in fact control the movement of substances into and out of the cell. And of course, we're only talking about plasma membranes here, but this applies equally to other biological membranes. So remember those membranes that surround organelles within the cell. So of course they also control movement of substances into and out of those organelles. Can anyone give me some examples of organelles that you might remember from our last lecture? You can remember an organelle, or in fact a structure that is not termed an organelle because it's not surrounded by a membrane. You can remember examples of organelles within cells. Mitochondria, good. Yes, mitochondria are organelles and they are found in both plant and animal cells. Chloroplast, yes, also an organelle, which of course is only found in plant cells. All right, what have we got here? Ribosomes. Ribosomes are found in plant and animal cells, but they are not organelles. They're not classified as organelles, but yes, they are very important structures. What else have we got? Golgi apparatus, correct? Both animals and plants, and they are termed organelles. It's interesting about the ribosomes, even though they're not, technically classified as organelles because they're not surrounded by membranes, they do carry out very important functions, and there is sort of some move towards actually rethinking this whole term organelle. So it could be that in the future ribosomes are termed organelles, and that whole definition of an organelle having to be surrounded by a membrane may actually change. All right, good. So we've got a few examples there. Also remember the nucleus, lysosomes, someone said Golgi. Oh yes, we've got Golgi. Endoplasmic reticulum is also another example. Okay, but let's move on with this lecture. Now, the permeability of molecules really depends on several factors. So first, the size of the molecule is important. So smaller molecules tend to be able to move through a membrane, but larger ones can't, and they need to actually travel through transport proteins that are embedded in the membrane. Electrical charge is also important. So these charged species, such as these ions that you can see here, tend not to be able to move through a membrane. Lipid solubility is also important. So remember, the interior of a membrane consists of those fatty acid tails. So we have this lipid interior, this hydrophobic lipid interior. So very small substances that are lipid soluble also tend to be able to move through membranes. But it really depends on all of those factors. All right, so an important concept here, diffusion. We're going to think about this term diffusion. And we can see the definition there, that diffusion is the passive movement of molecules along a concentration gradient. Also, we can say the movement of molecules down a concentration gradient, that is from a region of high concentration to a region of low concentration. But one of the important things to note here is that diffusion is a passive process. That is, you don't need to put any energy into the system in order for diffusion to occur. And a great way to think about this is to imagine a container of water. Oh, not very good. Container of water, and we're going to place a single drop of dye into that water. So of course this drop of dye represents a region of high concentration of that dye, and if you just leave it there, you don't agitate it, you don't stir it, you don't heat it up, you just leave it there, eventually those dye molecules will start to disperse, and they'll continue to disperse until they're evenly spread throughout that water. Can anyone tell me why those molecules, why the dye molecules actually move? Why are they dispersing through the water? So remember, we're not stirring it, we're simply placing that little drop of dye into the water. What causes them to move around? And it all goes back to our discussion of
does it have to do with like dissolving in like, and that's no
so that's the solubility, aspect. Yes, so the dye molecules are soluble in water, yeah, but I'm really after what's causing them to move. What do you think is actually causing them to move? That is, why do they actually disperse? Okay, electric charge, polarization, attaching to other molecules. Okay, so we're sort of on the right track, but not quite there. All right, think about kinetic energy. I'll give you a clue. Think about kinetic energy. Remember the energy of motion. So we're talking about dye molecules, we're talking about water molecules, and what do molecules always do? What are they always doing? A movement, exactly right. So yes, absolutely, molecules are always moving. They might not be moving very fast because there's so many of them in a liquid anyway, so many of them very close together, but they are always moving. And that movement means that the dye molecules, which are also moving, will eventually disperse throughout that sample of water. That's right. Okay, so that basically describes, really nice description there of diffusion, passive movement of molecules, so no input of energy. That energy is actually just simply provided because of that intrinsic energy that individual molecules already have. And the movement occurs from a region of high concentration through that sample of water to regions of low concentration. And you'll find that this really is a concept that's ubiquitous throughout the sciences. It almost seems to be a law of nature, that everything goes from high energy to low energy, or high concentration to low concentration. All right, so that brings us to diffusion of substances across membranes. Some substances can diffuse directly through the plasma membrane, through any membrane. They don't need any transport proteins. They can just move straight through the lipid bilayer. And here's some examples: oxygen, carbon dioxide, alcohol. These are all substances that can move directly across the membrane. But remember, they always move along or down this concentration gradient from high to low concentration. The rate at which they diffuse, those molecules diffuse, depends on how permeable, on the permeability of the membrane to those molecules, and it depends on the magnitude of the concentration gradient. So just thinking about this point here, the magnitude of the concentration gradient, let's imagine your plasma membrane, part of the plasma membrane of a cell, and I'm just drawing that as a lipid bilayer, without drawing in all the phospholipids because it would take me too long. And if you can imagine here, I know it sounds very simple. Oh, I can hear someone yawning. Is that you again, Simon? If you can imagine...
No.
If you can imagine a seesaw. This is a really good example. If you can imagine a seesaw where one end is completely up and the other end is completely down, and this represents a concentration gradient. So we've got a much greater magnitude here compared to another example where it's not quite as pronounced, that concentration gradient. Okay, so the magnitude of this concentration gradient is much greater, so diffusion, the rate of diffusion will also will be much higher. All right, let me clear that and move on. All right, so when it comes to ions, ions diffuse a little bit differently. They not only diffuse down a concentration gradient, but they also diffuse along an electrical gradient. And a combination of these two gradients is referred to as the electrochemical gradient. So the electrochemical gradient is a combination. Oh, I might have to mute someone there. Okay, so the electrochemical gradient is a combination of both the chemical gradient, that is the concentration from high to low, and the electrical gradient. And the electrical gradient is basically set up across the membrane because of this just a very slightly different charge that you'll find on both sides of the membrane. And remember that opposites attract and like charges will repel, but we'll come back to an example in just a minute. And the direction of net passive movement really depends on the strength of those two gradients. So here we have a nice example to illustrate what we're talking about. So in this top panel here, we're just looking at diffusion as a result of a concentration gradient. So let's imagine that this is a cell, and here are the particles, solute particles outside the cell. So high concentration here, low concentration inside the cell. So the direction of movement as we've said, when it comes to passive diffusion, is from high to low concentration. So those solutes will diffuse into the cell, and we can see now that we have approximately equal number of particles there, or concentration there, relative to the outside of the cell. So this is chemical equilibrium that's been reached. Now, when it comes to ions, which we can see at the bottom here, it's a little bit different. So let's take this cell once again, and we can see these negative charges just to the inside of the cell, and that's quite common for cells to maintain these very slight negative charges, and they're not charges that are found right through the actual cell. They tend to be localized just to the inside of the membrane. All right, we have a couple of questions before I move on. Does temperature affect diffusion? Yes, absolutely. Absolutely. So if you increase the temperature, obviously you're increasing the kinetic energy of the particles. They move faster, diffusion will occur faster. Does osmosis have a bearing on passive diffusion? All right, so osmosis, when we talk about osmosis, which we will later on, osmosis is a type of passive diffusion, but we'll talk more about that later on. All right, so to the outside of this cell, you can see these charged species, positive and negatively charged species, and you can imagine that because the interior of the cell bears a slight negative charge, which of these charged species will tend to move into the cell? Which ones would you imagine would move into the cell? Exactly, the positively charged species would be more likely to be transported into the cell, and you can see that here. So you've got mostly positively charged species inside the cell to balance those negative charges. And when this occurs, then we have achieved electrochemical equilibrium. So remember both the concentration aspect and the electrical aspect. Okay, good. Clear that. All right, so far we've been talking about, let me just write this here. All right, here's a membrane, lipid bilayer. We've been talking about diffusion straight through the membrane. Yep, diffusion straight through the membrane, passive diffusion from high concentration to low concentration. But often this type of diffusion is too slow to satisfy the requirements of the cell. So another type of transport that we'll be looking at here is often referred to as facilitated diffusion. And it's facilitated by these proteins that are embedded in the membrane, transport proteins, otherwise known as transmembrane proteins. Okay, so diffusion is still a passive process. There's still no input of energy, and diffusion, or sorry, movement across the membrane through these proteins still occurs from high concentration to low concentration. Okay. All right, so these transport proteins or membrane transporters or transmembrane proteins can be confusing when there's multiple names for the same thing. These membrane transporters accelerate the movement of less permeable molecules across membranes, or they can just accelerate the movement of molecules across the membrane, which need to be moved much quicker than they otherwise would when they're moving directly through the membrane itself. All right, now I need to just decrease the size of this a little bit. Oh, not that much. Hold on. Okay, that's better. So we can see this graph. All right, just looking at the graph down here, which is looking at the transport of glucose. So here we've got the rate of glucose uptake. And down along this axis, we've got the external concentration of glucose in millimolar. So this is the concentration of glucose outside the cell from 0 to 15 millimolar. Now, if we were just talking about this simple diffusion directly through the actual membrane itself, let's look at the rate. Here it is here, simple diffusion. It's quite low. It does increase as the external concentration of glucose increases. So glucose can move across the membrane, but very, very slowly. Now, what if we look at the rate of glucose movement across the membrane when glucose is moving through these membrane transport proteins? That is, membrane transporters that are specific for glucose. And you can easily see here how quickly the glucose is taken up into the cell. So this is referring to this type of transport. So still from high to low concentration, but facilitated through this membrane transporter. All right, so transport proteins tend to have very high specificity. Usually they're specific for one or a very small number of closely related solutes. But something else to notice here also in this graph is that even though the rate of transport increases, you can see that then it starts to level off. Why does it level off? The rate of transport across the membrane depends on the number of transport proteins that are situated in that membrane. If all of the proteins that are present are occupied, so they're all transporting, all fully occupied, transporting that substance, then that's when the rate levels off. I won't clear that drawing actually, I'll just leave that there if it doesn't interfere with something else. All right, so these transport proteins assist the movement of molecules down a concentration gradient through facilitated diffusion. Requires no energy input, still a passive process. There are two major types of membrane transport proteins. There are channels, which basically allow direct passage from one side of the membrane to the other. And then there's a different type, and these are called carriers. They tend to bind the solute on one side of the membrane. The act of binding will produce a conformational change in the shape of the protein, and this enables the solute to be passed from one side of the membrane to the other. All right, so let's talk about channels first. So these allow molecules to move in or out of cells very rapidly. They're faster than the carriers because they simply have these open and closed states, whereas the carriers have to bind, change shape, and then release solutes to the other side. So channels tend to transport ions, and they have very high specificity for specific ions. There's quite a few ion channels that have been characterised. We know of calcium channels, potassium channels, chloride channels, and there are quite a few others. All right, so this is what a channel protein would look like. So here's our membrane. Here's an example of a channel protein embedded in the membrane. This is in its closed state. And here we have another example, and here it is in its open state. So it's in its open state, then these molecules can simply pass through a pore within that channel protein. But remember, those solutes are moving once again from a region of high concentration to a region of low concentration. All right, and here we have the other major type of transport protein. These are the carrier proteins. So here's an example here of a carrier protein. And you can see that in this open state here, it binds the solute within this hydrophilic pocket. The binding of that solute, once the solute binds, that induces the conformational change. And you can see the change in shape here. And these proteins are known to be able to actually flip around, move around within the membrane. And you can see that happening here. It's flipped around, and that induces another conformational change in the actual carrier protein so that it opens to the other side of the membrane and releases the solute. So that change in shape means that it no longer has an affinity for the actual solutes, and they are therefore released. So they're our carrier proteins. Okay, back to the channels. So channels, as we've seen, have open and closed states, and they tend to be open and closed by various signals depending on the actual channel. So some channels are referred to as voltage gated channels. They respond to changes in voltage across the membranes. Then we have channels that are called ligand gated channels. They're activated by the binding of specific molecules, which are called ligands. And then we have mechanically gated channels, and these respond to physical disturbance. Okay, so now we move on to our third type of transport mechanism. And this type of transport is termed active transport. And let's draw this up here. So we use a transport protein once again. There we go. But this time, the difference is that rather than going down a gradient, a concentration gradient, we're actually going up a gradient from low to high. From low concentration to high concentration. So active transport requires energy, hence the term active. It requires an input of energy to move the solute against its concentration gradient or against its electrochemical gradient. And the molecule that's used most often to provide that energy is ATP. Who remembers this one? ATP. Okay, yes. Yep, few of you remember. Does anyone remember what ATP stands for? ATP. Something triphosphate, yes. Adenotriphosphate, very close, yes. Adenosine triphosphate, very good. All right, who remembers the structure of ATP? Who remembers the actual structure of ATP? So there's sort of three parts to it, something like that, like that. This is very rough, as you can imagine. Something like that. Who can fill in these circles for me? Tell me what these circles are. It looks like a nucleotide. Absolutely right. Yes. It looks very much like a nucleotide. Who can remember what these parts of ATP are? Who remembers these parts? A base, yes. So we've got a base up here. Great. And we've got a sugar. Yep, so I'll call that one S. And 3 phosphates. All right, look at that. 3 phosphates. All right, so next question to you. All right, what happens in order for this ATP to release energy? What happens in order for ATP to release energy? Something is broken. Break a phosphate group, absolutely. If we break one of these phosphate groups, then energy is released. So ATP becomes what? ATP becomes diphosphate, ADP, correct? Yep, because once you lose one of the phosphates, then we have diphosphate. And what if this ADP lost another phosphate? What if this bond here was broken? Then what would ADP become? And this doesn't happen very often. Exactly, mono. AMP. Okay. All right, good. That's terrific. All right, ATP. So let's look at active transport. All right, active transport employs the use of transport proteins, membrane transport proteins, just like our facilitated diffusion here. But these transport proteins with a difference. They are called ATPases, and that's because we require the hydrolysis of ATP to provide the energy for this transport protein to pump solutes against the concentration gradient or against the electrochemical gradient. And this is one example. This one is the calcium ATPase. So it's obviously specific for the movement of calcium ions. So that's one type of active transport protein. On the right here, we have another type. And this one is referred to as co-transport. Let's look at how that works. Let's start here in the middle with this membrane transporter. And this one is a proton ATPase, sorry. So once again, it uses energy from the hydrolysis of ATP to ADP. And that energy enables the proton ATPase to move protons against the concentration, the electrical and chemical gradient, so that the protons build up on one side of the membrane. Now, once you have a high concentration of protons on one side of the membrane, they will just naturally diffuse, if they can, they will naturally diffuse back down their electrical and the chemical gradient. So how can they diffuse back down that gradient? They can do that through additional membrane transporters. And this is one that allows protons to simply diffuse back down their electrochemical gradient. But this particular transporter here has two pores, one that allows the protons to diffuse back, and another pore that enables the transport of specifically amino acids. Now, what drives the movement of amino acids in the same direction through the same membrane transporter is the energy provided by this movement of protons. And this particular type of active transport protein here, which transports both the protons and the amino acids in the same direction across the membrane, is called a symporter, or simply symport. All right, so that's one type of co-transport. Let's look at the other side of this membrane here. Here we've got a different type of active transport protein here. So it still allows the protons to just diffuse, passively diffuse back down their electrochemical gradient. And that movement provides the energy for, in this case, sodium ions, but this time the sodium is moving in the opposite direction to the movement of the protons. So when you've got this type of co-transporter, where you've got one substance moving in one direction and something else moving in the other direction, it's called an antiporter, or simply antiport. All right, so that's co-transport. So co-transport can be described as diffusion of one molecule down its electrochemical gradient, that is the protons there and there, and that movement is used to pump a second molecule against its electrochemical gradient. All right, and this is a system that's used very commonly in plants. And we'll look at the system in animals in just a minute. Okay, so let me clear all that. All right, so let's just recap first. So in plant cells, protons are pumped from the cell by those proton ATPases, and this creates an electrochemical gradient for the inward movement of protons. That inward movement of protons provides the energy that drives the pumping of other solutes against their electrochemical gradients. But remember, the whole process is made possible because ATP actually enables the movement of protons against their electrochemical gradient in the first place. Now, in animal cells, there's a similar system, but it's the ATPase that's most commonly used is the sodium-potassium ATPase. And remember this image from our first slide, we're going to look at how potassium is kept at a high concentration inside the cell, low outside the cell, and for sodium, the concentration within the cell is kept at very low amounts compared to the outside of the cell. So, oh, here it is right here. All right, so in animal cells, sodium, potassium, ATPase, and we're going to look at a single nerve cell, but we'll just enlarge a tiny portion of the actual plasma membrane. And we'll just look at what's in this box here. All right, so here's the plasma membrane. This region here is the cytosol, so to the inside of the cell, and this region up here is the outside of the cell. All right, so here's our sodium potassium ATPase membrane transport protein. It utilises the energy from ATP. So this is showing ATP hydrolysis to produce ADP plus one inorganic phosphate that was lost from the ATP in the first place. And we can see what's going on here in terms of movement of ions. So we can see firstly that for every 3 sodium ions that are moved out of the cell, 2 potassium ions are brought into the cell. So this explains how animal cells maintain high concentration of potassium and low sodium within the actual cell. All right, now we can look at this part of the image, which actually shows what's happening in this process. So let's look at our sodium potassium ATPase here. So it's open, the opening is facing the cytosol of the cell, and this is where it binds 3 sodium ions. Okay, ATP is hydrolyzed to ADP, and the inorganic phosphate that's released, there it is there, that little yellow PI, that phosphate becomes covalently attached to the sodium potassium ATPase. And you might remember mention of this in one of our earlier lectures when we talked about that allosteric binding of a molecule to a protein, for example. And that binding can actually activate it or it can inhibit its action. So this is one example right here. All right, so when the phosphate covalently attaches to the sodium potassium ATPase, this induces a conformational change in the membrane protein, and you can see now that it's moved around, it's changed shape, and now those green sodium ions can be released to the outside of the cell. When they're released, 2 potassium ions can then bind, so they're shown as these little red triangles here. That will induce, once again, a conformational change back to the inside of the cell, so that it's facing the inside of the cell, and those potassiums are released, and the phosphate is also released. So another very neat little system that we can see there. All right, so we said earlier that cells tend to maintain a slight charge just to the inside of the membrane. And the difference in voltage across the membrane is called the membrane potential. And this can actually be measured in prac class, in fact. And we used to do that many, many years ago, measure membrane potentials. And I can tell you they do exist. All right, but not so much today. We don't have the very long pracs that we used to have 20 plus years ago. All right, so what sets up that membrane potential? The membrane potential is due to, firstly, the negative charge that we find on many proteins. Not all proteins, but many proteins carry an overall net charge. There's other polymers inside the cell that carry negative charges, but also the transport, the selective transport of ions across the plasma membrane. And if you measure that, that membrane potential, it's normally in the order of about negative 50 to negative 100 millivolts. Okay, also the removal of positive ions from the cell increases the negative charge inside the cell. Functions of membrane potentials. So membrane potentials provide favourable electrochemical gradients for the uptake of cations. The inward electrochemical gradient can be used in active transport of other ions, and membrane potentials or changes in membrane potentials can be used as signals. All right, now let's move on to a topic which I think, Simon, you mentioned this earlier, osmosis, the topic of osmosis. So osmosis is very much like diffusion. In fact, it's exactly like diffusion, except that osmosis refers to the movement of water itself rather than the movement of solutes, which is what we were referring to when we were discussing diffusion. So water travels passively across membranes. Water molecules somehow can move directly through membranes, which is quite unusual because they're very polar molecules, but they can move across membranes, and they do so by by osmosis. They move. Water molecules will move from a region of high free energy to a region of lower free energy. Let's look at what that all means. All right, this is a very important image here, which really describes osmosis and that concept of high and low free energy. All right, so here we have a sample of water of this side. Here we have in this dashed blue line represents a selectively permeable membrane. So this could be the plasma membrane or in fact any biological membrane. On this side of the membrane, we also have lots of water molecules, but we have here these large blue solute molecules. So these could be ions, sodium ions, for example, and chloride ions, but probably just one type. All right, so let's go back here. We've got only water molecules here. So pure water. There are no solutes, no ions, no other substances dissolved in this region here. And when you have pure water, sample of, hit the wrong thing there. Can everyone see the slide still? Oh, good. All right, because I hit stop share by accident. All right, when we have a sample of pure water, that sample is said to have the highest amount of free energy. That is, the movement of those individual water molecules is not impeded, it is not restricted by any other molecules that are present. Okay, so they have, those water molecules have the maximum amount of movement that you would expect. Okay, and this is given a value. It's given a value and it's set at 0 megapascals. That is the highest amount of free energy, set at 0 megapascals. So that means that any other sample of water that contains any solutes whatsoever will have a value that is less than 0. Less than 0, so they will all be negative values. All right, so free energy of 0. What about on this side here? What's happening here due to the presence of solute molecules? Can someone tell me what's happening? What is happening on that side of the membrane due to the presence of solute molecules? And you can clearly see hydrogen bonds with water molecules. It could be, depending on what those solutes are. Yes. Water molecules could be forming hydrogen bonds. What other type of bonds? So, all right, you can clearly see that those water molecules are actually surrounding the solutes. They are interacting with the solute molecules. What other type of bonds could they be forming? So hydrogen bonds is one. Yep. Yes. Dipole. We could have some iron dipole interactions going on there. Yeah. But the important point here is that because those water molecules are interacting with the solutes, their movement is restricted. That is, this sample here will have lower free energy than water molecules on their own, lower free energy. So whereas we've got a free energy value of 0 on this side, 0 megapascals, the free energy of water molecules on this side of the membrane might be something like negative 1.0 megapascals. Okay, so that's lower free energy on that side. All right, so as we said on the previous slide, and we'll just go back, as we said on the previous slide, water will travel from a region of higher free energy to lower free energy. So that means the direction of movement of those water molecules will be from this side of the membrane to that side of the membrane. That is from high free energy to low free energy. So the direction of net water flow will be from right to left. Okay, clear that. All right, let's go back one slide. Okay, so we can see already that the free energy levels are determined by the dissolved solutes. The higher the concentration of dissolved solutes, then the lower that free energy value will be. But there's another important point here. Free energy levels can also be affected by physical pressure, also referred to as tension. We'll come back to that in a minute. So the free energy of water is decreased by the presence of solutes, and it's also decreased when we have reduced pressure inside the cell. Okay, so let's recap. So water will flow across a membrane towards a region of highest solute concentration, because where the solute concentration is high, that means the free energy, the water free energy, is lower. And of course, in plant cells, pressure is also important because the cell walls are semi-rigid. But we'll come back to that in just a minute. Okay, so the overall free energy of water is referred to as the water potential. And here's a symbol for water potential. This symbol is called psi. And oh, I don't know why I keep hitting that button. Psi. Okay, there it is. So the overall free energy of water is referred to as the water potential. And the water potential is the sum of the solute potential or osmotic potential and also the pressure potential. Okay, so let's look at that again here. So the water potential, psi, is impacted by both the osmotic or solute potential plus the pressure potential. And this is something that you'll go into in a lot more detail when you study plant physiology in your second year. So we're only just introducing the concept here. Okay, so now we can actually define osmosis. Osmosis is the net passive movement of water from a region of high water potential to one of low water potential through a selectively permeable membrane. Okay, so what's that character for water potential? That character is called psi. The character for water potential, there it is, psi. It's called psi. I'm not sure if that's a Greek symbol. Can't quite remember, but it's called psi. All right, so let's look at now how this relates to different types of cells, whether we're talking about animal cells or plant cells. All right, so let's imagine that you took a cell and you placed it into some water. So there's our cell in some water. And the cell, the inside of the cell had a concentration of, let's say, 0.9%. And the solution that you placed it into also had a concentration of 0.9%. Okay, so that means the solution and the concentration inside the cell, they're exactly the same concentration, same osmotic potential. So we refer to that solution as an isoosmotic solution, also referred to as an isotonic solution. Just in case you see this word, which you would have seen if you could have done the prac that actually goes with this lecture. All right, same solute concentration, isoosmotic or isotonic. All right, now I'll just clear that or I'll run out of room. Now let's try again. And this time we're going to place our cell 0.9%, but we're going to place it into a solution that is 2%. So now this solution that we've placed our cell into is more concentrated, and we refer to it as a hyperosmotic or hypertonic solution. All right, the last one. Okay. Our solution, our cell, 0.9. And now we're going to place it into a solution that is only 0.1%. And of course, this solution is termed hypoosmotic or hypotonic because it's less concentrated than the solution within inside the cell. All right, now let's look at how different cell types respond to those different types of solutions. And we'll start with animal cells. We're looking at red blood cells here. So this is what a normal red blood cell would look like. And we can see the shape there, oval in shape with this depression. And when we place these cells into an isoosmotic solution, then the cells retain their shape and their structure. Okay, so let me just get this pen. All right, so once again, we're talking about a solution. So if we want to use an isoosmotic solution, then we would use about 0.9% sodium chloride. And that would be isoosmotic to the interior of the cell, about 0.85% sodium chloride solution. Okay, so that means that there's no net movement of water into or out of the cell. But what happens if we put red blood cells into a hyperosmotic solution? Here's our hyperosmotic solution. So this one is about 2% sodium chloride. Here's our cell. That's at 0.9%. Okay, what happens here? In which direction does the water move? In which direction does the water move? Moves into the cell. Okay, so remember, so looking at this again, where do we have the highest water potential? The highest water potential. Okay. Do we have the higher water potential? Or think about free energy. Is it higher here inside the cell or is it higher in solution? That's right. That's right. It's actually the water potential or the free energy of water is higher here because it's less concentrated inside the cell than it is in the solution. That's right. So the net movement of water will occur from inside the cell to the outside of the cell, that is into the solution. Yep. Absolutely right. And because water is moving out of the cell, those cells will shrivel, as you can see there. They will shrivel up And this is what you would see if you were actually looking at these cells through a microscope. Okay, so that process is called crenation, when the cells shrivel up like that. So remember, we're talking about animal cells here. All right, now what happens if we place the cells into a hypoosmotic solution? Okay, so this time, here's our cell, 0.9% concentration of solutes. We're going to place it into a hypoosmotic solution. Now, where do we have the highest water potential or highest free energy of water? The 0.1, correct. So this one, less concentrated here, so it has the highest free energy or highest water potential compared to the inside of the cell. So in which direction does water move? In which direction does water move? It moves into the cell, correct. So it will move from this, this region of higher free energy or higher water potential. Water will move into the actual cell. What happens to the cells? They start to swell. They're only surrounded by plasma membranes. So the cells will eventually burst. And this is called plasmolysis. Plasmolysis, lysis of the cell, breaking open of the cell. There we go. And this is what you would see under a microscope. You would see these cells that are swelling, and then you would also see some very odd-shaped cells after they've actually burst. Is this in animals? Yes, this is in animal cells. So of course, animal cells are only surrounded by membranes. So they tend to be very, very sensitive to the concentration of solutes in their extracellular environment. Okay, a little bit different for plant cells. Quite different, in fact. Okay, so let's start with this one in the middle. So we're placing a plant cell into an isoosmotic solution. Okay, and I don't need to draw. All right, so I might just clear that, sorry. Clear that and start again. So we place a plant cell into an isoosmotic solution. Okay, so let's say 0.9% outside the cell, 0.9% inside the cell. Okay, so same concentration, so there's no net movement of water in either direction. And for plant cells, this means that they remain flaccid. Okay, so very different to animal cells. Okay, so they don't shrink, they don't swell, they just remain flaccid, not the ideal state for a plant cell. Let's look at this over here. Now we're going to place the cell into a hyperosmotic solution. So 2% solution compared to our 0.9% inside the cell. Okay, in which direction does the water move when we put the plant cell into a hyperosmotic solution? In which direction does it move? That's right, it moves from the inside of the cell out into the solution. Correct. So what happens, we have the actual plasma membrane itself is shrinking away from that external cell wall. It shrinks away from the cell wall, okay, which maintains its shape, okay. And now if we go over here to the right and we place the plant cell in a hypoosmotic solution, in which direction does water move now? From the solution, yes, from the solution into the cell. Okay, so what happens, we can see the cell is starting to swell. Plasma membrane sits firm against the external cell wall. The cell wall also starts to swell. It starts to move outwards, but it eventually stops. It can only expand by about 10%, and then it stops, and then it starts to push back against the actual plasma membrane. And this causes a pressure to build up inside the cell, and that this is exactly the state that plant cells need to be in. So this is the desired turgid plant cell. And of course, you can imagine that that turgor pressure inside the cell is required in order for plants to maintain their structure. Okay. All right, so that means that plant cells, unlike animal cells, plant cells actually prefer or need to be surrounded by hypotonic solutions. Okay, because remember this was a hypotonic, and in fact they normally are, because plant cells or plants in fact are constantly transporting water through the actual body of the plant. All right, and I've probably gone into a little bit too much detail there. As I said, you'll get all this detail when you study plant physiology. Okay, so differences in the water potential across the plasma membrane of cells without cell walls, that is animal cells, are due only to differences in the osmotic potential, that is the concentration of the solutes. So that's for animal cells. But in cells that actually have cell walls, like plant cells, both the osmotic potential, that is the solute concentration, and the pressure contributes to the overall water potential, or psi. So water will enter the cell when the cells are placed in solution, in solutions that have less negative water potential. But the cells can only expand by about 10% before that internal pressure, that turgor pressure, causes the water potential within the cell to equal the water potential of the external solution. So it really is a combination of both the solute concentration and the pressure potential that contributes to the water potential. So of course, as we said, the shape of plants is maintained by that cell turgor. Water continually moves through the plant from the cells to the atmosphere, and the atmosphere has a very, very low water potential, somewhere in the order, depending depending on the day. The water potential of the atmosphere could be as low as negative 100 megapascals. Okay, so if that lost water is not replaced, then the pressure potential within the cell, and therefore the turgor, will be reduced. And then what happens? Plants will start to wilt. All right, so now let's move on to our very last type of transport. But we have a question. How many walls in a plant cell? How many walls in a plant cell plasma and membrane wall. Plasma and membrane wall. How many cell walls? Sorry, does that question, is that referring to how many cell walls in a plant cell? Plasma and membrane wall. How many cell walls has a membrane within it? Okay, so you're talking about the plasma membrane or the wall itself? The plasma membrane. So the plasma membrane is the actual membrane. That's the membrane that we're referring to, the plasma membrane. Plasma membrane has a membrane within it. The plasma membrane. Okay. So you're talking about this. About this when it's flaccid or when here. It doesn't matter either way. Either way, the plasma membrane is this line that you can see here. This is the plasma membrane, is the line. Yep. And this thicker green region here is referring to the cell wall, indicating the cell wall. Yep. And the same applies to this one on the right. So that line, this thin line here is the plasma membrane. Yep. That's fine. Okay, our very last type of transport, and this is called vesicle-mediated transport. And you remember we talked about vesicles last week in our lecture last week, and we saw that that vesicles are formed when small parts of the membrane surrounding the endoplasmic reticulum, small parts of the membrane butt off and form these little spherical vesicles, and they carry cargo to the Golgi apparatus, and the vesicle membrane fuses with the Golgi apparatus, thereby depositing its cargo into the Golgi, and then on the other side of the Golgi the same thing happens again. So larger molecules are transported in these membrane enclosed vesicles. The process where cells actually bring substances into the cell is called endocytosis. And this is basically facilitated by the plasma membrane itself. The plasma membrane will enclose substances outside the cell, and then that small portion of the plasma membrane will pinch off to the inside of the cell to form one of these little spherical vesicles. And so it's importing substances to the inside. When the vesicle is carrying solids, the endocytosis process is called phagocytosis. If the cell is importing fluids, then it's referred to as pinocytosis. All right, so let's look at this on the next slide. All right, so endocytosis, bringing substances into the cell, transporting larger substances usually into the cell, but not always. All right, so this first image at the top here actually is showing something that the cell is not intentionally doing, but it is showing the way that some bacteria, so these are bacterial cells here, and it shows that how some bacteria can actually infect host cells. They basically, they can either trick the cell into starting to curve inwards. The plasma membrane is curving inwards. This is called invagination of the plasma membrane. Here the invagination continues around the bacterial cell, and here the invagination is nearly complete, and the plasma membrane is about to pinch off here to form this vesicle. This vesicle which has internalized the bacterial cell. All right, so there's various ways that bacteria can do this, but some of them can simply trick the host cell into thinking that it's bringing various substances into the cell. Other bacteria simply produce some really toxic, what are called virulence factors that can degrade the membrane and gain entry into the host cells in that way. But if you look at this image on the right here, this is an actual image of a bacterial cell invading the plasma membrane of a cell. It's been coloured, pseudo-coloured, and this would have been taken, this image would have been taken using a transmission electron microscope. And you can see the scale bar here at 1 micron or 1 micrometer. All right, here's another example. So this middle panel here. Okay, question, is that a plant cell? I can't really tell. I don't believe so. I don't believe it is. It looks like an animal cell from what I can see. I can't see a cell wall in this image here at all. All right, so here's another example. Once again, we've got solutes outside the cell. Invagination of the membrane is taking place. Eventually, that invagination is nearly complete. It will pinch off that little bit of plasma membrane and internalise the solutes. So you can see that every time endocytosis takes place, a little bit of plasma membrane, that external plasma membrane, is taken into the cell. Yeah, so we lose a little bit of plasma membrane every time that endocytosis takes place. And there is something that the cell does to counteract that loss of membrane. But here's another example here. And this time, we're looking at a part of the plasma membrane where we have these blue proteins embedded into the membrane, and these proteins happen to be what are called receptor proteins. And receptor proteins will bind to specific target molecules that you can see here in green, and only when the right target molecules bind to the receptor proteins, this actually triggers plasma membrane invagination, and then finally that internalizing of that piece of the plasma membrane to form a vesicle, which is carrying the target molecules to the inside. And of course, to the right, you can see an actual image of that taking place. Transmission electron micrograph, which is quite amazing. And look at the actual scale bar there, 0.093 microns from what I can see. All right, and here's the opposite type of transport. So this is called exocytosis. This type of vesicle-mediated transport. And this is where the vesicles are actually formed within the cell. So much like the process that we just mentioned before, these vesicles may have come from the Golgi apparatus, and they're carrying substances that are destined to be secreted from the cell. So the vesicle approaches the plasma membrane. Here at the plasma membrane, the membrane of the vesicle itself will fuse with the plasma membrane of the cell. You can see that that's actually occurred here. When it fuses with the plasma membrane, it opens up, the membrane opens up, and the secretory products are released from the cell. So in this way, we have extra pieces of membrane that are being added to the plasma membrane. So that counteracts what we saw on the previous slide, where small regions of membrane were being lost as they were being internalised. All right, so in summary, membranes control movement into and out of cells. Some movement across membranes is passive. So we saw just passive diffusion, and we saw facilitated diffusion. That is diffusion that's facilitated by transport proteins. We also saw active transport, which requires energy mostly provided by ATP. Water almost always moves passively across the membrane. Although they have discovered membrane proteins called aquaporins that seem to be specific for transporting water across membranes. That's another topic. Okay, and larger molecules can be transported across the plasma membrane by vesicle-mediated transport, which we just saw for endocytosis and exocytosis. All right, that's the end of this lecture. So is it time for a break? I think it is. Time for a break before we move on. So shall we break, let's say for 5 minutes? Is that enough? Let's make it 7 minutes. Enough time for a cup of tea. All right, in that case, we need 10 minutes. Cup of tea. Okay, sounds very good to me. All right, I'll be back very soon. All right, who's got their cup of tea? Or coffee? Yes, I have my coffee. I'll have my tea just before I go to bed. Right, have I still got that record on? Yes. Green tea, very good. Yes, I need to start on the green tea again. It's very refreshing. Oh no, the record is still going. Excellent. All right, so still on the topic now of, yes, caffeine. I can still sleep, doesn't matter how much caffeine I have. I think I'm very immune to caffeine. All right, so we're still talking about cells, but now we're going to look a little bit at tissues, and we'll start this lecture by looking at animal cells for those students that are studying animals, and then we'll finish with a look at plant cells and tissues. So this is just an introduction for you. And let's consider first an embryo. Whether it's a plant embryo or an animal embryo, that early embryo is just a mass of cells, undifferentiated cells. That is, they're all the same. They don't have any specific function as yet. And then very early on during the development of the embryo, some of those cells will start to take on a specific form and function. That is, those cells are said to differentiate, and those cells will continue to differentiate both through animal and plant development. Then similar cells will come together to form tissues, and then different tissues will combine to form organs. And in complex animals and plants, organs will combine to form systems. So, of course, something like the respiratory system in an animal or the transport system in plants. All right, so we're going to start by talking about animal cells. Now, with animal cells, the plasma membranes of adjacent cells may be separated from each other, and they're separated by what's called an extracellular matrix, which we like to abbreviate to the ECM, extracellular matrix. The ECM is a fluid network of proteins found in a hydrated polysaccharide gel, and it's secreted by certain cell types. All right, the extracellular matrix contains many different proteins. These may be structural proteins or they may be adhesive proteins. The two major types of structural proteins are collagen and elastin. So if you imagine and do it right now, take the back of your hand and pull up the skin from the back of your hand. It's the actual elastin, the elastin proteins that actually allow you to stretch your skin up from the back of your hand. But it's the actual collagen proteins that prevents you from ripping the skin off the back of your hand. So it's the collagen proteins that have very, very high tensile strength. They have very low elasticity, but very high tensile strength. And in fact, it's been predicted that gram for gram, collagen fibers have higher tensile strength than steel. That's amazing. Gram for gram, collagen fibers have higher tensile strength than steel. That's incredible. Elastin proteins, on the other hand, elastin proteins have very low tensile strength but very high elasticity. So they're the proteins that allow you to actually stretch your skin. All All right, then we have various adhesive proteins, fibronectin, laminin, and others. All right, the major polysaccharide that we find in the extracellular matrix is called the glycosaminoglycan. So this is a large polysaccharide that's bound to a protein core. It consists of strong negative charges. So these negative charges attract cations into the extracellular matrix, and because we have cations moving into the ECM, water will naturally follow by osmosis. So that means that you have a very highly hydrated network, very highly hydrated, and that high degree of hydration actually provides cushioning against compressive forces. Okay, the proteins that are found within the extracellular matrix integrate cell activities and guide cell movements, but that's not something that we'll talk about today. There are two major types of ECM. One is called the interstitial matrix, which is found in connective tissues, and the other type is called the basal lamina, which tends to be found under epithelial cell layers. And we'll have a look at that on this slide here. All right, so to the left, we have some upper epithelial cells that you can see up here. Underneath that, we have this very tough sheet of basal lamina. Remember, that's one type of extracellular matrix. And then under the basal lamina, we have cells of the connective tissue, and those cells are surrounded by the other type of extracellular matrix, which is called the interstitial extracellular matrix. All right, that's also illustrated for you here on the right, probably easier to work out what's what. So let's start from the top. So we've got a single layer of cells here. These are epithelial cells. Underlying the epithelial cells, we have the basal lamina, which is that shaded region there. And then underneath that, we have the connective tissue. And within the connective tissue, we can see some cells. So here we have an odd-shaped cell, which is called a fibroblast cell, and these are the cells that actually secrete the extracellular matrix itself. Here we have a macrophage, a mast cell, so these are just cells of the immune system, and we have lots of extracellular matrix in between those cells. Oh, and of course a capillary, a blood capillary. All right, here we have some collagen fibers, and we have some elastin fibers here, these red fibers. And here we have those large polysaccharides, these fibers here, called glycosaminoglycans. All right, so where cells are in close contact with each other, so we're still talking about animals here, the plasma membranes of those adjacent cells are connected. They're connected by what are called junctions. These are molecular complexes that connect cell to cell or connect cell to the extracellular matrix. There are three major types of junctions, the tight or occluding junctions, the anchoring junctions, and the communicating or gap junctions. And we'll look at each of these in turn. Okay, but before we do, let's look at all of them together. So what we're seeing here, so in the middle, we have one cell of the intestine, And to the left, we have part of an adjacent cell, and to the right in yellow, we have part of another cell. All right, so let's look at all these different types of junctions that are illustrated here. So here, in between these two cells, we have what's referred to as a tight junction. Is another type of junction that we can see here called an adherens junction. This is a type of anchoring junction. Another type of anchoring junction here. Here we have a communicating junction, otherwise known as a gap junction. There's another example there to the inside of the cell. And another type of anchoring junction at the bottom here. Okay, we can also see to the inside of the cell that some of these junctions are actually connected to components of the cytoskeleton. So remember last week we talked about that internal scaffolding, if you like, within the cell. The cytoskeleton, and there's three major components of the cytoskeleton. So these green fibres that we can see here, these are filaments of the cytoskeleton, and then these brown fibres that are connecting junctions to the inside of the cell, they are also components of the cytoskeleton. All right, and at the bottom here, we have one type of extracellular matrix, which is called the basal lamina. So let's start with the tight or occluding junctions. These are junctions that prevent the free movement of molecules through the extracellular fluid in between cells. So this is pretty important in certain parts of the animal body. And we revisit a cell here of the intestine. So here's the lumen of the gut, so the inside of the gut here. And we can see that we've got some glucose within the lumen, and that glucose has to be transported into these cells. So the glucose is, firstly, it's actively transported into this cell. So we can't see the transport proteins, but there are lots of transport proteins on this side of the cell, in this upper part of the cell. Transport proteins that are specific for glucose. Okay, so glucose is taken into the cell against its concentration gradient. Concentration of glucose builds up inside the cell, and then glucose can actually diffuse through transporters here, just passive transporters through this side of the actual cell, this lower part of the cell, into the bloodstream. Okay, but the important point here is that the tight junctions that are found in between adjacent cells prevents glucose from moving in between the cells. It forces the glucose molecules to move through the cells, into and out of those cells. Another important function of the tight junctions is that they restrict the different membrane proteins, those different glucose transporters, to specific areas of the plasma membrane. So remember we said that there, in this upper part of the intestinal cell, there are active transport proteins for glucose, but in the lower part of the cell, there are passive glucose transport proteins. And that one of the jobs of the tight junctions is to actually limit where those or control where those transport proteins are actually found. So that's the tight junctions. Now we move on to anchoring junctions. So these provide mechanical support. There's different types of anchoring junctions. As you saw on one of the previous slides, there are desmosomes, there are hemidesmosomes, and there's different types of adherens junctions. All right, and we can see some examples here. So here's part of one cell in yellow and part of an adjacent cell down here. And so we can see the this particular type of anchoring junction is anchoring one cell to the neighbouring cell. And then within each of the cells, the molecular complex of that particular junction is linked to part of the cytoskeleton within the cell. Okay, so these fibres are actin fibres. Right, and here's another type of anchoring junction here. So in purple, we have the actual, the complex, the molecular complex of the junction. And in this case, it's anchoring this cell here to the extracellular matrix itself. Okay, and this part that you can see here, this is one of the proteins within the extracellular matrix. Okay, so we've seen those anchoring junctions here in this image. All right, and finally our communicating or gap junctions, and these are specialised for electrical and chemical communication between cells. And we can see some examples down here. So these are the regular protein channels that we can see on one side of the plasma membrane of one cell. And here's the plasma membrane of an adjacent cell. And we can see here how those communicating junctions actually extend from one cell to the next. Now moving on to animal tissues, and we're going to start with epithelial tissue. So epithelial tissues form continuous layers as surfaces, not just surfaces on the surface of the animal itself, but also lining any internal cavities. Epithelial cells are bound together by tight and anchoring junctions, and they function in protection, regulation of exchange of materials, and secretion. Epithelia are categorised by either the number of cell layers or by the shape of the cells. So if there's only one layer of cells, the epithelia is called simple epithelia. If there's more than one layer of cells, it's referred to as stratified epithelia. When we look at the shape of epithelial cells, flattened cells are called squamous epithelia. Cube-shaped cells, cuboidal. Column-shaped cells, columnar epithelial. All right, this table shows the different types of epithelial tissue. So starting with our simple epithelium. Okay, and we start with squamous, simple squamous epithelial tissue. We can see a nice example there. So these are very thin cells, flattened cells, and this provides a thin layer across which diffusion can readily occur. So of course you would find squamous, simple squamous epithelial cells lining the lungs. Next one, simple cuboidal epithelium. So obviously a thicker cell type, so still only one layer of cells, and these are typically found lining some glands. These are cells that are rich in specific transport channels, and they function in secretion and absorption. Next type, simple columnar epithelium. And we can see once again, single layer of cells, but they are columnar in shape. And these tend to be found lining the stomach and the intestines. So we saw these very long column-shaped cells in the gut on one of the previous slides. So these are thicker cell layers which provide protection and also function in secretion and absorption. All right, next we move on to the stratified epithelium. So more than one layer of cells. So here we have our squamous stratified epithelium, typically found in the outer layer of skin and lining of the mouth, a tough layer of cells which provides protection, and then our last epithelial tissue, this is the stratified or pseudostratified columnar epithelial, which tends to be found lining parts of the respiratory tract. There are specialised epithelial cells within this tissue which actually secrete mucus, lots of mucus. And many of these cells also have these little projections. They look like little hairs, but they are actually called cilia. There's the word there. I don't know if you can read that. C-I-L-I-A, cilia. So, of course, the function of the mucus that's secreted by these cells is to trap any foreign particles, whether those particles are dirt, bacteria, virus particles. The mucus traps those foreign particles, and then the movement, the sweeping movement of the cilia actually moves that mucus with all its debris up to the back of the mouth, where it can be swallowed. Okay, so that's epithelial tissue. Our next major tissue type in animals is the connective tissue, which provides basic structural, metabolic and defensive support. So connective tissue normally has a large amount of extracellular matrix, ECM, that we were talking about earlier, and that ECM may be calcified, as you would find in bone and teeth. It can be fluid, as you would find in blood. Okay, so the components of connective tissue, so normally the ECM, the extracellular matrix, is more abundant than the cells themselves. And we know that the ECM is a matrix of polysaccharides and proteins. The fibres that we find in the ECM, we already know about collagen, we know about elastin, and here's another example, reticulum. Okay, to the right, we have this table from your textbook that describes the major types of connective tissue. So we've got loose connective tissue, dense connective tissue, cartilage, bone, and blood, and some location, typical location and functions, which you don't need to remember, but they are there for your interest. Our next major tissue type in animals are muscle tissue. So, of course, muscle cells are capable of contraction. They contain lots of actin and myosin filaments. And muscle tissue, of course, is responsible for movement of the animal, but also for movement of internal organs. Muscle tissue is very broadly categorised as either striated or smooth muscle. Striated muscle is highly organised. For example, skeletal and cardiac muscle, which is illustrated here. Here's your skeletal muscle and cardiac muscle. And then, of course, we have smooth muscle, which has a less regular arrangement than striated muscle. And this is muscle that you will normally find around your internal organs. Our final tissue type in animals is the nervous tissue. So nerve cells, which are known as neurons, will carry information throughout the body of the animal. let's have a look at the structure of a neuron. So we can see an actual image here. So there's one neuron there and a second neuron there. That's also illustrated here. One neuron and the second neuron there. All right, so let's look at the different parts to a nerve cell, to a neuron. All right, so this is the main part of the cell here, which is called the cell body. Then we have all these projections from the actual cell body, and those projections are referred to as dendrites. Then we have a very long projection from the cell body, and this is called the axon. The axon itself is supported by several different cells. Several cells, and these are called glial cells. Glial cells, and the glial cells will form a very important protective sheath around the axon called the myelin sheath. All right, so that's it for our animal tissues, and we have some examples here of where those different tissue types may be found within the body of an animal. Now moving on to plant cells. So unlike animal cells, plant cells of course have rigid cell walls, and this limits both their size, limits their shape, and of course those plant cell walls tend to be very closely connected to each other. So they tend not to have that extracellular matrix that we see in connective tissues and other tissues in animals. All right, so let's have a look at this image here to the right. So we have one or part of one cell up here. And this darker line that you can see is the plasma membrane of that cell. Here's an adjacent cell with its plasma membrane. And then in between them, this lighter grey mass that you can see is represents the cell walls of both of those cells. We've seen in the previous lecture that the presence of a rigid cell wall or semi-rigid cell wall allows plant cells to be surrounded by a hypotonic solution. That is a solution that has a lower concentration of solutes than the concentration of solutes that you would find inside the plant cell. So without that semi-rigid cell wall, the inflow of water from that hypotonic solution by osmosis would cause plant cells to burst. But of course that doesn't happen because the cell walls prevent cells from lysing. Cell walls are also important in plant metabolism. They contain lots of enzymes, they act as a pathway for transport, absorption, secretion, and they act as a barrier against pathogens. In this image here to the right, you can actually see this is the outside of a plant cell wall. That's how it looks with the aid of an electron microscope. Let's take a look at the structure of a plant cell wall. Cell walls of plants are composed of cellulose, the structural polysaccharide called cellulose, and you might remember the monomer unit of cellulose. The building block of cellulose is glucose. So cellulose is composed of chains of glucose molecules linked together and organised into microfibrils. Those microfibrils are then embedded in and linked to a matrix of other polysaccharides. one of which is called pectin, and various proteins. So let's have a look at that down here, and I'll just enlarge that so we can see that properly. Oh, move that over. Oh. All right, so now we can see this a little bit better. So in green, we have our cellulose microfibrils. So 1, 2, 3 are shown here. Then we have 2 different types of pectin molecules. We have acidic pectin molecules, so here in red, and then we have these smaller neutral pectin molecules in yellow. In purple, we have glycoproteins, and we have different types of polysaccharides, which are indicated here as non-cellulosic polysaccharides here attached to the cellulose microfibrils. Okay, so that's basically the structure of the cell wall. Okay, I need to make that a bit smaller. There we go. All right, so let's look at the development. Oh, have I moved slide? Oh, I have. Sorry about that. Okay, so what about cell walls of fungi? Cell walls of fungi, they're a little bit different. These cell walls are composed of a substance called chitin, and chitin is very similar to cellulose. It's composed of subunits that are similar to glucose, but they're not glucose. And we saw that in one of our previous lectures. So the main component of chitin, that is the building blocks, is N-acetyl glucosamine, which, as you can imagine, we abbreviate to NAG, NAG, N-acetyl glucosamine. All right, what about cell walls of bacteria? They're composed of 2 different polysaccharides, one of which is N-acetyl glucosamine. And those 2 polysaccharides, those 2, not polysaccharides, they're monosaccharides, they're linked to a chain of 4 amino acids. So cell walls of bacteria are quite different. Include that. All right, let's take a brief look at the development of cell walls. So let me just draw a single plant cell, and this plant cell is getting ready to divide. It's getting ready to undergo what's called cell division and divide into 2 cells. And we won't talk about cell division until next semester, but when it starts preparing to divide, it first assembles a new region of plasma membrane. 2 new regions of plasma membrane, okay, start to form, because this one cell here is going to divide into 2 new cells. So first the plasma membrane, the new plasma membranes form, and then the wall material, the new cell wall material, is going to be formed to the inside there, in between the plasma membranes. And the way it does this is that it transports glucose precursors into that area and then the glucose precursors are assembled into cellulose within that region to form the new cell wall. And that new cell wall that's formed is called the primary cell wall. Primary cell wall. So this is quite a thin, quite flexible cell wall. Later on, many plant cells will form a secondary cell wall, and the secondary cell wall is quite unique and different to the primary cell walls because it contains a very tough substance called lignin. So I'll just clear that. Okay, so as we said earlier, plant cells tend to be quite closely connected to each other. And the rigid cell wall actually limits contact between the adjacent cells, but the cells are connected, and they're connected via these very fine little tubes that you can see here in between these cells. And those little tubes are called plasmodesmata. Plasmodesmata are channels that link plasma membranes and the cytosol of adjacent cells. So the plasmodesmata are actually continuous with the plasma membrane of this cell and with the plasma membrane of the adjacent cell. Okay, so the plasmodesmata itself is surrounded by plasma membrane. Now within the plasmodesmata, there is another tube to the inside, and that tube is called a desmotubule. It runs through most plasmodesmata, and the desmotubule is continuous with the endoplasmic reticulum of both cells. So we not only have a connection from one cell to the next via the plasma membrane and the plasmodesmata, but we have another connection which is not shown here, and that is via the desmotubule that runs along the inside of the plasmodesmata. All right, now we're going to move on to plant tissues. And unlike animals that have 4 major tissue types, plants have 3 major tissue types. But before we look at plant tissues, we're going to look at the most important growing regions of a plant. So let's take a look at this plant down here. Oh, and I don't know why I can't see. Just put that up a little bit. Oh, here we go. Okay. All right, so at the very tip of a growing shoot, we have a specialised region of active growth called the shoot apical meristem, and that's abbreviated to SAM, the shoot apical meristem. So that's found at the very tip of the growing shoot. And there's another one down here. So just behind the actual root tip, there's another region, specialised region of cells, and it's called the root apical meristem, or RAM. So they're easy to remember, SAM and RAM. All right, so what's important about these regions here? So if you think about stem cells in animals, embryonic stem cells is a good idea. You probably already know that stem cells are undifferentiated cells, and when they do differentiate, they can differentiate into virtually any cell type. That's exactly what we have here in these meristems, these apical meristems, the SAM and the RAM. And we can see an electron microscope. This is a scanning electron microscope image of a shoot apical meristem. So you couldn't see this with the naked eye. So the shoot apical meristem is a cluster of cells located about here, just a small cluster of cells. The number of cells really depends on the plant species. And these are cells that are undifferentiated and they're continually dividing to produce new cells. They're continually dividing. So one cell will divide into two cells, and these two new cells that form are called daughter cells. In each pair of those daughter cells, one of those daughter cells will remain undifferentiated. So it will remain as a meristem cell. The second of those daughter cells will differentiate into part of the mature body of the plant. So it might start differentiating, for example, to form a leaf. It might start differentiating to form a lateral bud or a flower. But remember that there's always one daughter cell that remains undifferentiated, so that meristem persists throughout the lifetime of the plant. And the same thing occurs in the root apical meristem down here. All right, so those meristem cells have actually been mapped. So let's say that's the region of meristematic cells here. They've actually been mapped. That is, they've actually discovered that there are specific meristem cells here that will give rise to specific tissue types. So dermal meristem cells, I'm not sure exactly where they're found, I don't remember, will give rise or differentiate into dermal tissue. Ground meristem cells will differentiate into ground tissue, and vascular meristem cells will differentiate into vascular tissue. So let's look at those three tissue types. All right, starting with our dermal tissue, and I'll just make that a little bit smaller. Oh, that's better. Right, dermal tissue forms the outer covering of a plant. And epidermis, the epidermis is the major type of dermal tissue. It consists of either one or several layers of closely packed cells, depending on what part of the plant we're looking at, and it is highly specialised. Epidermal cells are not photosynthetic cells. They don't contain, or most of them don't contain chloroplasts. They don't photosynthesize, but they are very highly specialised. And one of their major functions is to secrete this waterproof layer over the surface of the plant body, and that layer is called the cuticle. The cuticle is made up mostly of this lipid type substance called cutin. So we can have a look at this image down here. So we're just looking at a very small portion of a leaf, which has been enlarged here. And this outer cell layer, this is the epidermis, a single layer of cells in this case. And the epidermal cells are secreting a layer called the cuticle. So not a layer of cells, just a layer of cutin. All right, so that's the epidermis, and below the epidermis you can see different cell types. These are the ones that are specialised to photosynthesize. All right, so the function of the primary function of the cuticle is to prevent water loss to the atmosphere. But the plant still needs to take in carbon dioxide, and it needs to be able to remove water vapour from the inside of the plant leaf. So in order to facilitate that, the epidermis actually contains tiny pores called stomata, and that's not shown anywhere here, but those tiny pores are surrounded by two specialised epidermal cells called guard cells. And those guard cells, those two guard cells, will control the size of that stomatal pore opening. And of course, it's through these stomatal pores that the exchange of gases actually takes place. And we also have other specialised epidermal cells. So here we can see some epidermal cells that have leaf hairs, glands, and these are also specialised in elongated epidermal cells that form what are called trichomes. All right, so that's our dermal tissue. Now let's move on to tissue type number 2, ground tissue. Ground tissue includes storage tissue, structural tissue, but of course, most importantly, it includes a tissue involved in photosynthesis. And our final tissue type, the vascular tissue. So vascular tissue is the tissue that transports water, minerals, and synthesized carbohydrates throughout the body of the plant. There are two major types of vascular tissue in plants. One type is called the xylem and one is the phloem. So of course the xylem vascular tissue is responsible for transporting water and minerals which are taken up from the soil solution by the roots. And the water and minerals are transported always in an upwards direction towards the leaves. So that's the xylem vascular tissue. And then we have a very closely associated type of tissue called the phloem. And this tissue transports photosynthetic products, that is carbohydrates, predominantly from leaves, to places of use or storage. So the phloem will transport those products of photosynthesis in all directions, in an upward direction, downwards through the plant, and also from side to side. All right, in this image here, we can see part of a plant. Looks like the stem here and maybe some lateral branches. And these red and green lines actually represents the xylem and phloem. Okay, if we cut a section through that stem, here it is here, so we can see the inside of the stem. We can see these little coloured regions here, which are referred to as vascular bundles. And within each vascular bundle, if we take one of these vascular bundles and enlarge it, we can see the vascular tissue within. So here we have the xylem, these red cells of the xylem, and to the right we have cells of the phloem. So they're always very closely associated with each other for very good reason, which we won't talk about here. But that is the all-important vascular tissue. So in summary, in multicellular organisms, cells are specialised and often linked. In animals, we have 4 major types of tissue: epithelial, muscular, connective and nervous, whereas in plants, well, in vascular plants, there are 3 major tissue systems: dermal tissue, ground tissue and vascular tissue. That completes our lectures for tonight. How's everyone holding up there? Did the coffee work or the tea? Thank you, everyone.
Savannah, I just had a question about our final exam.
Yes, final exam. All right, bear with me. Hold on. Hold on, because I've got something to show you.
Okay.
All right, so I haven't released this exam timetable yet. because it's still a draft and I still need some confirmation. But yes, ask away while everyone's having a look at this draft timetable.
Just in regards to doing it online,
Yes.
will we still be required to like draw cells and cell structures?
No.
Okay.
No, definitely not.
Sad. Fun.
It's too hard to draw anything when you're
Yeah.
doing an online exam. Yeah, no, definitely
Okay.
not. No. But we can see here that most likely the Living Systems exam will be on Monday, June the 15th. All right, let me just make that a bit bigger. All right, so here we go. All right, so the Living Systems exam is 1.5 hours. But because it's going online, in fact, all the final exams are going online. As you can see here, that's all been approved. And because it's going on, because they're going online, we need to give you more time to complete those exams. So the general rule is to add 50%. And then split those exams into 2 shorter exams. And that's why you can see that for Living Systems, instead of one single 1.5 hour exam, you'll be having 2 70-minute exams. So, so you've got that extra time because it's always slower doing exams online, always slower. You have to click between each question. You know, your internet might be running a little bit slowly. You don't have the benefit of the reading time. It's always much faster when you're doing an exam using a hard copy in class. You'll also see that for all the exams, all the final exams, you will have a 24-hour window in which to complete the exams. 24 or nearly 24-hour window in which to complete the exams. So although the exams are timed, that is, once you start an exam, you'll have a certain time to finish it. But you can start those exams at any time between 12 a.m. and 11:59 p.m. on the scheduled day, and that will apply to all the final exams.
Savannah,
Does that make sense? Yes.
Does that mean that you could do one exam on the same day for 70 minutes?
Yes.
And then have a rest and then do the other exam for 70 minutes?
Exactly right.
Okay, so you could do...
Exactly right. So you will do, you could do part one of the Living Systems exam, because you can see there are two exams. You could do part one at 10 o'clock in the morning,
Yeah,
and then you can do part 2 at 6 o'clock in the evening, if you like.
I see.
Yeah.
So even 3 a.m. in the morning, I guess.
You could do it at 3 a.m. if you like.
Okay.
And the reason this has been introduced is because there's been, well, obviously there's, problems with the, due to the virus, people are at home, kids are at home.
Yeah.
It might not be ideal for you to be doing an exam at a specified time with no flexibility. So that's why we've introduced the 24-hour windows for all the exams.
Okay.
Okay.
What
So does that make sense to everyone?
Yes, it does.
yes.
Oh, someone's Mitch, is this bizarre? Are you talking about the exams are bizarre?
Yeah, it's just, being able to choose when you can do it and not be under the eye of the
Of the invigilator.
yeah, it's just it's very unusual.
It is very unusual, but these are very unusual times.
Absolutely, yeah.
Very unusual times, and you can see that La Trobe and of course Melbourne Polytechnic, we're trying to be as considerate and as flexible as we can possibly be, given these very unusual times. These are all open book exams, all open book. But even though they're open book, this does not mean that students can work together. Okay, so keep that in mind, that students should not be colluding. Because so be very careful. There will be in the instructions, you'll see that students that are suspected of colluding, and sometimes it's really obvious when they are. So students are working together. If students, oh, excuse me, if students are suspected of colluding, they will be required to attend an oral defence. Okay, so make sure, you know, it is open book, but you must do it on your own. You must do the exam on your own.
Just a question about the two separate exams.
Yes.
One be predominantly chemistry focused and the other biology focused, or just be a blend throughout?
It will be a blend.
Okay.
It will be a blend. Yeah. Yeah. So I haven't quite finalised how I'm going to split the exam, but I'm fairly sure that I'll have all the multiple choice in one exam, and then there'll be your true and false short answer and some calculations in the other exam. I'm fairly sure that's how I'll be splitting the exams. Okay, will the exam be covering all topics throughout the semester? No. The exam will cover everything except for what we covered in the mid-semester test.
So week 7 onwards or whatever.
Yes, that's right. That's right, from week 7 onwards. Yep.
Cool. Silvana?
Yes.
May I ask a question about the lecture?
Sure.
I didn't know which part of the lecture the question would fit into.
Right.
It might be thinking a bit too far ahead, but I was at work today and we were discussing how plants, when they grow, they come into contact with like a solid structure.
Yes.
Like with trees and their root, they'll contort their growth so that they don't grow straight into a wall or whatever.
Yes, they certainly do.
What factor sort of like makes that happen at a cellular level? Is it like, because it's not a physical one, because they don't crash into it and then change direction? Is it like what's the?
That's right.
Yeah.
It's more a sense. It's more that they're receiving some signal for that there's an object present.
Would it be pressure, like a pressure thing on the cells?
Oh, that's a really good question. That's a really good question. And
You mean like grapevines?
so look, there's been a lot of studies done on exactly what you're talking about, and studies that were done right back to when Charles Darwin was actually researching that very phenomenon. And and I can't quite remember the mechanism that they've come up with. That avoidance mechanism.
Because espalier and grapevine trellis systems kind of make sense because it's like a physical manipulation of the plant growth, or it's like facilitating it in a certain direction.
That's
Whereas
right.
with trees, they kind of just grow out and do all these weird contortions and yeah.
They certainly do, yeah.
So no, that's okay.
That's a really good question. Yeah, and there are mechanisms that have been determined for that to actually explain what you're talking about, but it's not something that I can quite remember.
No, that'll be a good YouTube
Yes,
rabbit hole
yes,
procrastination
absolutely.
session for me. So that's good.
Yes, and can you share with me when you find it?
Yeah, we'll see
And
where we end up.
Let's
Yeah.
see where we end up. And I'm pretty sure that I've actually got a video that explains that. I'll have to. Now you're going to make me go back through all my files.
Can
to
you
find
put
that video.
if you find that, could you just put it
Yes.
up on Moodle or something?
Yeah, absolutely.
That'd be sick. All right, cool.
Absolutely. Yeah, no, that's really interesting.
All right, thanks for that. Thanks for the
No,
lecture.
no problem. You're welcome. You're very welcome.
Silvana, it's Luke.
Yes. Hello, Luke.
Hi. Would I just be able to ask a question about agriculture systems and tech?
Sure. I don't know if I can help, but I'll try.
No, that's okay. Yeah, I did email Doug, but I
Right.
just want to keep bothering him because but, yeah, if because our final mark might be withheld because we can't do the chemical users.
Correct.
course.
It will be withheld. Your final mark will definitely be withheld. Yeah.
Okay, because I guess a large portion of that subject was 7 weeks at Yang Yen doing those courses. Are we now required to do like the extra work, which was like these extra online quizzes for that subject, or will
No, not at all. Those assessments, which are the tractor operations course and the chemical users course, those assessments have not been replaced.
Okay.
So those assessments will stand, and they will be delivered when we're allowed to deliver them. So all students that are doing Ag Systems and Tech, your final. You'll be placed on a withheld at the end of semester, and then we just simply have to wait until we're allowed to reschedule those short courses.
And what do you mean by that? So like in semester 2, we'll just have to try and fit it in with the rest of the course load, or?
I don't think that's probably not going to work. To do it in semester 2, because that means that students may be overloaded.
Yeah.
It might work for some students. Some students might be happy to do that. So we're going to have to be very careful and very flexible with when we schedule, when we do the scheduling. So I suspect that we'll probably, put out some tentative dates to reschedule those courses. But then, of course, if that doesn't suit all students, then we might have to consider running those courses outside of semester. So, for example, closer to the end of the year.
Yeah.
Yeah.
Okay.
Yeah. So, that's a really good point there. Because the last thing we want to do is get back into, the full semester of full-time study for some students, and then burden them with additional stuff that they couldn't do in first semester.
Yeah.
Yeah, that would be really tough and unfair.
Okay.
Yeah.
Cool. Thanks.
Yeah. Okay, no problem.
Savannah, I just had another question about similar to doing those practical courses.
Yes.
Practical elements for this subject, like all of our pracs and things that we can't really learn through video.
Yes, for living systems.
Living systems.
Yes.
And winemaking.
Yes.
Will we be trying to do those again as well? Specifically asking about the forklift license or training.
Forklift. Yeah, I don't know about Is anyone doing forklift?
Well, I don't know if it was actually
it was included in the winemaking. It was in part of the schedule for the semester that we would be learning about forklift operations. Right.
I actually wasn't aware of that you were doing forklift as part of winemaking.
Yeah, it said to be confirmed in our schedule, but it was definitely in there.
Right, okay.
Which was definitely enticing me into the subject.
Oh, right, okay.
Yeah.
Oh, that's interesting, because the forklift...
I don't know if it was a license, but it was definitely like operational.
Yeah, no, my understanding that the forklift course wasn't running this year. So did you talk to someone about that, or you saw that written somewhere?
I tried to ask. It's written in our in that document, like on the first page of every subject on Moodle.
Right.
It's written in there. It says to be confirmed, but I asked Mark about it, and he said just due to Corona, he wasn't sure what was going to happen.
Right.
But I just
okay.
haven't heard anything else about it.
Yeah,
And
the forklift training, that's normally organised through Ag Systems and Tech, through the Ag Systems and Tech subject, along with your tractor driving and the chemical users. But I'm almost positive that that's not running this year, and that's why it hasn't been incorporated as it usually does. It usually is, yeah. Yeah, but in
will
terms
it be incorporated into something later on?
not to my knowledge, no. No, I'm not even sure that they're still running the forklift course at all.
Okay.
Yeah, I'll have to look into that. That's one for Doug. Question for Doug, because he used to organise the forklift course. But yeah, I'll see what I can find out about that, about the forklift training. In terms of other practical skills, what we're planning to do for some subject is to offer catch-up pracs later on in the year. So they'll be optional catch-up pracs. So students don't, necessarily have to attend, but if they wanted to, experience some of the practical skills that they've missed out on, then they'll certainly be able to do so. Some of those catch-up pracs will be incorporated into subjects in such a way as so you're not actually doing a lot of extra work, if any. Other catch-up prac sessions will be organised outside of semester. All right. Any other questions for me?
Doctor, I have a question about the lab prac 1.
Yes.
I'm not sure if it's okay to ask this question.
Yeah.
But when we talk about references, I'm having difficulty putting enough references. How many are you really after? Because at the end of the day, it's 500 words.
That's right. Yeah. So
I can't see provision for having too many references.
You can't have too many references. No, not at all.
Right.
Yeah. So normally students in a 500 word prac report, you might have, say, 2-3 references.
Right.
Yeah, that would be enough.
I see.
2 or 3 references.
Right.
So you'll definitely need a couple of references in your introduction.
Yep.
And then you might use, 1 or 2 references in your discussion.
Yeah.
Yeah.
Okay.
Yeah, so you certainly don't need too many references at all.
Mm-hmm. And the other question I have, in the Excel spreadsheet, when you put your data on the standard curve,
yes,
there is a formula, y equals mx plus b, for instance, yeah?
Yes, yeah.
Now, there is an optional video that explains how you can do that.
Yes, there is. There is. Can you see that on Moodle?
Yes, I can.
Yeah.
I just wanted to understand, is it compulsory to have that equation